Next comes peptide linkage. The structural organization of proteins is complex and is composed of alpha amino acids joined by peptide bonds. A peptide is a condensation product of alpha amino acids. For example, the condensation of glycine and alanine is represented here. The COOH group of glycine and the NH2 group of alanine condense. One molecule of water is eliminated and forms dipeptide glycyl alanine. It is written as gly-ala. In the structure of the dipeptide, there is a CONH linkage. This CONH linkage is called peptide bond. In a tripeptide, 3 amino acids condense together to form the tripeptide chain. The structure of a tripeptide is represented here, in which the first one is an alanine unit, second is a glycine unit, and third one is a phenylalanine unit. The COH group of alanine condenses with the NH2 group of glycine and forms a CONH peptide bond. The COH group of glycine condenses with the NH2 group of phenylalanine to form the second peptide bond. And finally we get the tripeptide ala-gly-phe. The first one is alanine unit, second one is glycine unit, and the third one is phenylalanine unit. By convention, while writing the structure of peptides, the free amino group is represented on the left side and the free COH group is represented on the right side. The condensation product from a large number of amino acids is called polypeptides. And proteins are formed from polypeptides. Generally, proteins have 100 or more amino acid units in their molecule. Based on the structure and solubility in water, proteins are classified into fibrous proteins and globular proteins. Fibrous proteins are thread-like. They are insoluble in water. Examples include keratin in hair, skin, nail, etc. Fibroin in silk, myosin in muscles. Globular proteins have spherical structure They are soluble in water. Examples include hemoglobin, insulin, etc. The figure shows collagen, which is a fibrous protein, and hemoglobin, which is an example for globular protein. Next comes structure of proteins. The structure of proteins are considered in four different levels. They are primary structure, secondary structure, tertiary structure, and quaternary structure. The specific sequence in which various amino acids are arranged in a protein is called the primary structure of that protein. The primary structure decides the biological activity of that particular protein. Hemoglobin contains 574 amino acid units arranged in a specific sequence. The conformation which the polypeptide chains assume as a result of intramolecular or intermolecular hydrogen bonding is called the secondary structure of that particular protein. The two common types of secondary structures are alpha helix and beta pleated. The figure shows alpha helix and beta pleated structures. In alpha helix structure, the polypeptide chain coils into a spiral form due to the formation of intramolecular hydrogen bonding. The CO group forms hydrogen bond with the NH group of the same polypeptide chain. Example in keratin, myosin, etc. The secondary structure is alpha helix. In beta pleated structure, there is intermolecular hydrogen bonding. The polypeptide chains lie side by side in a zigzag manner and it forms hydrogen bonding, intermolecular hydrogen bonding to give flat sheet-like structures. The silk protein fibroin has a beta pleated structure. The three-dimensional structure of a protein which arises due to further folding and superimposition of different secondary structural units in a characteristic manner is called the tertiary structure of a protein. The tertiary structure gives rise to two major molecular shapes, the fibrous and the globular shaped proteins. Hydrogen bonds, ionic or electrostatic bonds, van der Waals bonds, disulfide bonds, weak hydrophobic interactions, etc. come into play during the formation of tertiary structure of a protein. Several proteins are composed of two or more polypeptide chains, and these polypeptide chains are called subunits. The spatial arrangement of these subunits with respect to each other is called the quaternary structure of that protein. For example, hemoglobin has four globular polypeptide tertiary structures combined together to give the quaternary structure of the entire globular protein. The forces that stabilize the quaternary structure include hydrogen bonds, van der Waals forces, disulfide linkages, electrostatic forces of attraction, etc. At a normal pH and temperature, each protein will take a shape that is energetically more stable. And it is called the native state of that particular protein. Changes in temperature, pressure, pH, presence of salts, certain chemical reagents, etc. can disrupt the complex three-dimensional structure of the protein. This is referred to as the denaturation of protein. The disruption of the native conformation of a protein by an external agent is called denaturation. Denaturation causes a protein to lose its biological activity. It changes the secondary and tertiary structures of the protein. The primary structure of the protein is not changed by denaturation, but the secondary and tertiary structures are disturbed by denaturation. Example, hard boiling of egg. The soluble globular protein albumin present in the egg white undergoes denaturation into fibrous form and it gets coagulated when the temperature is increased. Another example is coagulation of milk. When milk is heated with an acid, example lemon juice, the globular milk protein lactoalbumin undergoes denaturation into fibrous form and gets coagulated to form cheese.